Hi and welcome to some updates I've just been making to Manic Microbes. So it's now got a bit of a UI. You can load and save slides, which are saved games, and create a new one. I still need to put some actual settings you can change against the simulation, but just dumps you into a new templated scenario at the minute. You can still add sort of substrate blocks to this to sort of separate out and partition different areas, and you can play around with the light level, and the efficiency of chlorophyll is really just a way of controlling the total energy input into the system to control some of the growth, if you want to keep it within sensible numbers. But I've added a sort of an oscillation to the light as well, so you can simulate a day-night cycle. And you can play around with the speed, and just sort of tap spacebar to pause and think, if you want to be able to investigate things, and then unpause and it all keeps going. And then so some of the other things that have changed in here, well, there's now sort of tags against these objects to represent species and genus labels that essentially you get because I store an exemplar version of this DNA against a species or a genus, and then I measure the genetic distance from that exemplar by a little calculation that does sort of longest continuous sequence, comparing two different sort of DNA sets, and then if it's beyond a sort of threshold, it decides, no, that's a different species or a different genus, and yeah, I just create a new one and make that specimen the new exemplar for that new species. And so there's a whole family tree I want to be able to sort of show graphically that would be really good to see, and then you can trace back how many individuals are in those different stages and how things have branched out, and then go back and have a look at the changes. And this changes because when mutations happen through when they reproduce, then you can either have sort of point mutations to this code, or a sort of a section of it can be deleted, added, removed, reversed, those kind of changes. And yeah, because this is quite ordered in terms of being put in a particular order to do a particular function, any kind of random change is going to increase the entropy and sort of probably break what this is. And if it breaks it in a significant way, it'll sort of just not function and die, and that's the whole point. So it's only the successful ones that don't break this, or either don't make it worse or potentially make it better, will be the ones that carry on reproducing. And so that's what I hope to be able to sort of show. This is a very similar thing to the way Darwin bots worked, and that worked. I think one of the considerations to make in sort of designing this language, though, is just to make it very flexible and not very fragile, so that you can change this in any number of ways, and it's not like it sort of breaks and can't run at all or can't compile. It is just commands running against a value stack and an address stack, and then it sort of puts the result back on a stack, or it writes a value into memory, which is this sort of big buffer of memory here. And so really all it's trying to do is orchestrate the sort of creation of these organelles, and it's the organelles that then do something in particular, sensory movement with the cilia, or and in fact even to the fact that there's a sort of membrane and a ribosome. If you remove the membrane, it's basically committing suicide. It will just delete the cell as it spills out. But that's the sort of job of this DNA, is to construct it. At some point I'll put a sort of resource management system to some extent in, so as well as just energy, you've got sort of this concept of nutrients that might just be the sort of amino acids that are needed to build some more complicated proteins that you use to then build the actual content of the cell. And then there's a couple of extra things in here before they get eaten. So this is an example cell for showing the neural net. It's literally just got 6 neurons in here, an input and output for hidden neurons. And I've just I've wired them up here and showed sort of two different ways of wiring them up. So there's some default commands to wire in or wire up. Wire in just wires to the layer above, or sorry, to the input layer explicitly. Wire up will wire to the layer above. Maybe let's put the light oscillation on, because I can see these all spreading out of control a little bit. So obviously without light, a couple of things happen. These no longer get so much energy. In fact, they'll lose energy while the light's off, because it costs energy to each of these organelles costs energy to run, some small amount. But with the light on, obviously the chloroplast organelles will make energy. So long as the light's coming on and off, they'll be fine. If it stays permanently off, they'll all die off. The orange hunter cells just have a bit of logic that randomly tries to turn around, and if it sees something within the wavelength of 500 to 540, it will just run straight towards it and try and eat it. And if it can't, it'll turn around, and that's literally all it does. And that's nice and simple. As you can see, when the light's off, they can't see, so they just sit here, not doing anything at all. And then when the light comes back on again, they'll be able to see, so then they go back to doing what they're doing. These yellow ones aren't sort of written in the same way, so there's a neural net behind what they do, so they're slowly learning. In fact, these actually seem to be doing something. There was a couple of these spotted round, and of course most of the time they sit there not doing anything at all, because they just sort of start off with a random jumbled set of weights and biases. But yeah, back to this one. So this has this really basic neural net, and then I just write to the memory locations of 40 and 41 the contents of what it can see with the light sensor and what the final output neuron says, which are these two values here. And past 200 seconds of being alive, which it is now, it will just jump straight to here, which is to set the cost element of the neural net to 0. And that is because, I've set variable 45 to just be the cost for all neurons. I could have set them independently and in various other ways, but they're just all set to 45, and 45 gets set to 0. So this value here is 0, so it's not currently training it in any way. So this is just based off of the weights and biases it's already had. Although, to be fair, it probably didn't learn very well, because I had the light on permanently for the entire time I was demonstrating. So I'll probably need to retrain this one. You could do that by just setting this gene number 1 that's currently set to run once, and so there's a -1 in there that says it's deactivated. You could set that back to 0 or a positive number, and this would run around again, essentially reset all the weights and biases. It'd probably need to reproduce to give it a new age, and then it would sort of potentially relearn. But this will then show an output value between -999 and +999, depending on whether the light's over 2000 or under 2000. Is a sort of simplistic example. And so these are interesting, because obviously the orange ones aren't eating themselves or anything else other than green. So it's only if they're facing one of these green ones will their code activate the sort of mouth to try and ingest food, whereas the yellow ones really just have a range of output neurons set to control their the cilia in the mouth, so their movement and whether or not they eat is just totally controlled by the outputs of the neural net. So as I say, normally you just get sort of fairly random behavior until some of these weights and biases start to get trained by the training code down here, and the training code down here just sort of says something simple. This is if 120 would be the mouth, so if it's ingested something recently, it gets a reward. 101 will be the distance from the light sensor. So if something is very close in front of the light sensor, that will get a reward. Although -1, it looks like so I'm inverting the distance to give it a reward. So and then 450 is the energy, so if it's under 450 it's going to get the cost applied to it knows it's doing something wrong, and then as soon as it will go above 450 there's no cost applied. So those are sort of training weights that all go to make up the value that I put in 40 or 41. Yeah, okay. And then that's what's used to train this neural net. And it's just a continual sort of training loop, essentially. As in the example of the other one, you could have it where it eventually switches off training, but there's no real need. And the state of these organelles, when it reproduces, at the moment it just clones absolutely everything and all the data in it. What I want to do is there's commands to give the ability to duplicate these, and then a split command will split wherever the or you set a split command on a membrane. So if it's at the minute I just set a value in 9 and it totally duplicates completely, whereas I'll change that pretty soon to be it splits from this membrane organelle down, so you can pick what goes in and potentially make some kind of egg or seed or virus or other kind of things where you could potentially make a new cell that just had a few specialized organelles and a membrane, and then you set it loose, and that's basically, as long as it's got one membrane in it, it'll survive. If it doesn't, it just, the contents spill out and it dies. So long as it has a ribosome, it will run DNA code. If it doesn't, you can make a virus where it just had some DNA code and it had some organelles, but it didn't actively run it. Not sure about this. This is getting a bit probably not to the right level of abstraction I was really after, so I may rethink quite how this works. But yeah, and then there's some sort of nice syntax highlighting in the DNA code, so you can tell when you've got a command correct, and I label out the genes. I mean, these genes, as you can see, aren't hard. There's no hard and fast way of doing this. These are just tags. And gene start just runs as a sort of bit of syntactical sugar for if the value behind tag number 1, which is memory location 1, if it's positive, run; if it's negative, skip. And where does it skip? It'll skip to the next instance of 1, which is at the end, and so it stops. And then gene stop just knows how to set this value. So stop literally will set it to -1, so it'll only run through the once. End just clears out the tag that's in the so we've written the tag here. End or stop will clear this out from the tag stack, so that it's not the current layer of tag anymore, because you can see you do things like this. Jump command is working on the basis that we're in tag number 3. We set tag 3, so tag 3 is still the valid tag when we call jump, so it'll jump to the end here, so you can use it for some branching logic. And then there's another example of that where I've put a tag 5 here, so if this cell goes below 120 energy, we figure we're not doing very well, so jump to 5, which rewires up all these neurons. I think there was some code originally in there to set the random weights and biases, which is really what I was after. Yeah, okay. So obviously the longer you leave this, and if you save this and come back to it, these will slowly learn, and as they reproduce, they'll pass those learned traits down to their offspring. And then hopefully this will be an interesting interplay between the sort of instincts, I guess, of the learned weights and biases from the neural net itself, and the code that generated and set up the neural net and trained the neural net. And so both can be changed, and so this could be mutated in a way that trains it differently or sets it up and wires it differently, and then that'll be interesting to see how well, or what that, which has the biggest impact. And yeah, that's how it's going so far. Okay, I think that's it. Okay, bye.